We left off last time in the course talking about co-translational import, all of the processes that happen when mRNA is being translated, starting at the rough endoplasmic reticulum, through all of the different steps that could be involved to translocating that mRNA or the protein product into the lumen of the ER, and the post-translational modifications that occur in the lumen of the ER, the lumen of the Golgi, along each one of the different stacks of that organelle, trafficking it to the plasma membrane. Today we'll begin with the other type of protein trafficking, which we call post-translational import, involved with the localization of proteins that are translated in the cytosol and then routed to specific locations in the cell. I have a couple slides dedicated to nuclear localization sequences and other sequences that regulate nuclear versus cytoplasmic localization of proteins. Mitochondrial targeting sequences are also an example of post-translational import, not quite as dynamic as the processes that I'll describe to you on the next couple of slides, but similar in that they're encoded in a linear amino acid sequence of proteins after they've been translated. Nuclear localization signals, or NLSs, in fact, engage in an entire network of proteins involved in shuttling cargo in and out of the nucleus. We're going to lean heavily on the cartoon shown on the right of this slide here to give a high-level overview of what the driving forces are to move cargo. And when I refer to cargo, it's the protein that has the NLS or some other sequence on it into the nucleus or out of the nucleus. As I said before, NLS-containing proteins are synthesized by ribosomes free in the cytosol of the cells. And then after the protein has fully translated, it can get routed to the nucleus as follows. To orient you on the slide here, to be able to move an NLS-containing protein into the nucleus, we need to move up a driving force, because if this protein is localized, concentrated in the nucleus, each time a new protein is synthesized, it's got to go up the concentration gradient to move where the higher nuclear localized protein that's already been made. That's thermodynamically unfavorable. The way in which the cell circumvents that thermodynamic challenge is to use another thermodynamic gradient to make it favorable inside the cell. And the other gradient that counterbalances the concentration gradient for this NLS-containing protein involves a small G protein, GTP binding and GTP hydrolyzing protein called RAN. NLS proteins will go up their concentration gradient as follows if there's a strong enough downward gradient of RAN going from the nucleus into the cytoplasm. With that context, I'll walk through the pathway here. After an NLS-containing protein has been translated, the nuclear localization is signal is exposed and able to be recognized by other proteins. And the family of proteins that recognize nuclear localization signals are called importins. Importin, there are several subunits of importance, which is going to focus on the family. They recognize the NLS-containing protein, bind to it, and it's the importin NLS-containing protein that is moved together as a complex through this pore called the nuclear pore complex that are large holes in the nuclear envelope that enable selective transport of cargo in and out of it, provided that the protein is large enough to be filtered by the nuclear pore complex, or NPC. Importins will, once bound to cargo, will readily move through the nuclear pore complex. Once they do, this importin NLS complex encounters GTP-loaded RAN, and this drives the dissociation of the importin NLS complex, and then it has the importin bound to RAN GTP. So it's the dissociation, and now importin is in is in the nucleus by itself. Now, to enable important to get back into the cytoplasm to bind additional NLS-containing cargo, this RAN important complex will go down the concentration gradient of RAN GTP from the nucleus back through the nuclear pore and into the cytoplasm. It's in the cytoplasm that the GTPase activity, and there are in fact binding proteins that promote the GTP hydrolysis that's bound to RAN, will hydrolyze GTP into GDP. This dissociates the exported importin in the cytoplasm, and now we've closed the loop. That free importin is available to bind additional cargo, and you cycle as follows. There are also circumstances where a protein may need to be excluded from the nucleus. First question might be, why? You just said it was hard to get into the nucleus. It shouldn't be hard to keep it out. Well, what happens if you have a small protein, something smaller than 40 kilodaltons, which, as I said before, will diffuse passively in and out of the nucleus? The NPC doesn't have the ability to block out that cargo. Yet it may be crucial to have this protein be exclusively localized to the cytosol. In addition, there are multiple circumstances of transcription of factors, I don't have to give you any more details than that, that have both an NLS and one of these other signals to move that protein out of the nucleus in a regulated fashion. So there could be post-translational modifications that obscure or reveal certain targeting sequences, enabling dynamic regulation between the cytoplasm and the nucleus. And that export-mediated regulation is encoded by another signal that occurs post-translationally. It's in the primary sequence of the protein, and that's called a nuclear export sequence, or an NES. So NLSs drive proteins into the nucleus, NESs ship them out. And the binding events that occur with NESs are thematically similar to the binding processes that occur with NLSs. Many of the steps, though, occur in reverse, and they involve other family members in this broader family of proteins that shuttle back and forth to the nucleus. We will begin in the nucleus for this cartoon. If there's a protein that's in the nucleus that has one of these NESs, it forms a complex with, rather than an importin, with an exportin, and together with RAN GTP in this heterotrimeric complex, gets exported out of the nucleus. Again, running down, basically hitching a ride, if you will, on the RAN GTP gradient from inside the nucleus to outside of it. Goes outside, and all this stuff is the same. There's a protein that binds here, activates the hydrolysis of the GTP, and upon GTP hydrolysis, the complex dissembles, and then the exportin is brought back into the nucleus for another round of export. The past two slides had these nice barreled cartoons of what the nuclear pore complex looks like. We in fact have great structural detail on the NPC. This is not even the most recent example of this is still blobology. We have crystallographic information on all of the subunits in the assembly of the NPC. It is enormous. MDA, that's megadaltons, 50 million molecular weight. And it is a self-assembly of many different proteins that interact with one another in different symmetries to form the pore itself. And the proteins that self-assemble and bind to and help form the nuclear pore complex are called nucleoporins, or NUPs for short. And there are several dozens of these. The exact numbers differ if you're talking about yeast or you're talking about humans, vertebrates, and invertebrates. But if you take an inventory on this, this is several hundred individual protein species, complexes, and subcomplexes. Some of them are crucial for establishing the structure of the NPC. How do you get the ring? How do you get the pore? There are brush-like extensions in the center of the NPC that provide the gating function as to whether or not large proteins go in or out of the nucleus. And then there are also proteins on the inner ring and the outer ring of the NPC that determine interactions with proteins such as exportins and importins to allow the cargo to move in and out. So it's a whole field of biology that people's entire career is on, just the nuclear pore. We're going to move on now to another category of post-translational modifications. Didn't fit neatly into those subheadings from before, but will enable us to get back to Noonan syndrome at the beginning of this lecture. And these relate to another category of post-translational modifications that occur to different ends of a protein after it has been translated that change the biomolecular character of that protein to localize it biophysically to membranes, in essence. The first category that we'll discuss is a hydrophobic modification called prenylation. And what a prenyl group are these hydrocarbons here, double bond, single bond, and then a branch here. Prenylations occur on proteins. They'll occur on any protein which at its C-terminus has what's called a CAX box. And CAX is nothing more than the one-letter abbreviation for the amino acid sequence at the C-terminus that will render it prenylated. C is a cysteine, A, two alanines, X is any amino acid. If there's a protein that has CAA something, it will get prenylated. If you want to prenylate a protein artificially, all you have to do is add CAAX at the C-terminus and it will undergo this modification. The prenylation modification occurring on the C-terminus creates a hydrophobic group on that C-terminus that makes it thermodynamically favorable to wedge that prenylated protein into the inner leaflet of the plasma membrane. There are different forms of prenylation that are defined by the number of prenyl groups that are concatenated together. If there are two prenyl groups, it's called geranyllation. If you have three prenyl groups, it's called farnesylation. And heaven forbid you should have four prenyl groups, because then it's geranyllation. And we'll come back to those in a moment because they're relevant to some of the Wrasse MAP kinase signaling that I introduced before. There are other modifications that I want to mention for completeness and also to get a sense that it's not just the ones that we're going to dive into greater detail are. There are also lipid modifications that will tether proteins to the outer leaflet of the plasma membrane. The major category are anchoring to glycophosphatidylinositol, thankfully called GPI. GPI-linked proteins is another C-terminal modification that will create a covalent bond between glycophosphatidylinositol, a component of cell membranes of the plasma membrane. And if there's a covalent conjugation between that and the C-terminus of the protein, that protein is going to remain on the outer leaflet of the cell unless there's some sort of lipase to chop it off. And then the final modification to be aware of is called a myristylation. It is the conjugation of a myristoyl group now to the N-terminus of a protein. These modifications localize proteins to the inner leaflet of the plasma membrane, and the myristylation motif that drives myristylation is different. Methionine, the start methionine, and a serine. If you have methionine, serine is a candidate for myristylation. And we talked about Wrasse last time, and we said how Wrasse is on the plasma membrane all the time, just like the heterotrimeric G proteins. Wrasse is on the plasma membrane because it is farnesylated. G alpha, G beta gamma, the heterotrimeric, I think they're geranylated or geranylated. And this is the reason why they're on the plasma membrane. When prenylation occurs on that CAX box, the cysteine plays a critically important role. There are enzymes that do these conjugation events. I'm not going to dive into those. But the essence of that enzyme catalyzed reaction is that the free thiol on that cysteine now creates a thioether bond with the prenyl groups, farnesylated, geranylated, et cetera. And that creates a stable covalent linkage that will place that prenylated protein on the plasma membrane as follows. It wedges right in there with the long chain aliphatics of phospholipids. Myristylation, thermodynamically, pretty much the same thing. The chemical substrate that's conjugated to the N-terminus of the protein is different. It's myristic acid. But at the end of bringing that myristoyl group to the N-terminus, catalyzed by the indicated enzyme on the slide, you now have this hydrophobic prong on the N-terminus of the protein that is most likely to go to the inner leaf of the plasma membrane to be most thermodynamically stable. And it's the myristylation pathway and the modification that will enable us to circle back to Noonan syndrome and the Wrasse MAP kinase cascade. And the reason is that in a subset of individuals with Noonan syndrome, there is a specific mutation in a scaffold protein, more to come on that in a moment, called SHOC2. And we'll speak about what the mutation is in a moment, but I want to say what scaffold proteins do. We introduced at a high level the Wrasse MAP kinase pathway, the gestalt of it, Wrasse to RAF, MEK and ERK, and kinase to kinase, kinase to kinase. Wrasse, as I mentioned, because of its farnesylation, is at the plasma membrane. And it needs to be near the plasma membrane to interact with receptors to become GTP loaded to begin with. RAF, the first kinase in the cascade, is not a plasma membrane localized protein. It's in the cytosol, but it can be recruited to the plasma membrane. It can be perchance near the plasma membrane so that Wrasse can activate it. So Wrasse will allosterically activate RAF on its own, but the interaction of Wrasse and RAF can be aided if there's a third protein that binds each of them, and by binding each of them helps the two proteins to interact more effectively with one another. This is the essence of a scaffolding function. And I'm going to draw some things on the board here because there's oftentimes confusion between adapter proteins, when we talked about clathrin-coated pits and other adapter type of processes, and distinguishing that from scaffolds, because they both appear to bind to other different things, but the terminology conveys a certain meaning. Adapters, as you saw in the earlier slides, bring co-localize two different proteins, but those two proteins don't necessarily need to have any chemical interaction with one another. And so if there's a protein A and it's located somewhere, and there's a protein C that you want to localize to protein A, you can get an adapter B that'll bring them together, and now you've localized C where A is by virtue of B. Yes? Thank you. Better? A scaffolding protein can enhance an interaction that will already happen on its own, but by binding the two players that would ordinarily interact, it increases the local concentration of those two interactors, and accelerates the rate or stoichiometry of the reaction. Often the way that these are drawn is now let's say that A will react with C. You can put those two in a test tube and the reaction will occur. But if you add in B, something like this here. You have two pockets, one for A, one for C. Now A and C are much closer to one another. Increased local concentration, if this is an enzyme-mediated cascade, huge increases that can occur. Now, interestingly, if there's too much B, you actually can inhibit the reaction because you could sequester A in one complex by itself and sequester C in another complex by itself. So there's some interesting modeling and things that can be done about the optimal amounts of a scaffold to promote signal transduction. But this is the key difference between adapters and scaffolds. And shock 2 is a scaffold that brings together Wrasse GTP and RAF. As I already said, RAF is in the cytoplasm ordinarily. Shock 2 is in the cytoplasm ordinarily. RAF is at the plasma membrane. If shock 2 can help RAF interact with Wrasse at the plasma membrane by binding it in this way, and then bringing RAF to the plasma membrane, that can improve signal transduction through this pathway. In a subset of Noonan syndrome patients, shock 2, they have a mutation in shock 2 that changes position 2 of the translated protein, which should ordinarily be a serine, to a glycine. Recall that the first position is always a methionine, and now we have methionine glycine on shock 2. Now, shock 2 has a motif for myristylation. In papers that I'm not showing you, will show that it gets myristylated. And now, shock 2, in addition to Wrasse, is localized to the inner leaflet of the plasma membrane. Two different post-translational modifications are giving rise to them that go to the inner leaflet of the plasma membrane, but now they're both there. When they're both there, because shock 2 can also bind RAF effectively and access that scaffold, the net result is now RAF is almost constitutively to the plasma membrane. And chemical reactions, binding events happen very fast, much more rapidly on a surface than they do in solution, like in the cytoplasm. Thus, aberrant myristylation of shock 2 gives rise to chronically activated signaling through the MAP kinase pathway. And as we said in the introductory slide, this chronic activation of MAP kinase is chronically changing gene expression. And for individuals that have an inherited mutation in this scaffolding protein, this is occurring all during development, right? All during the life of these individuals. And this has impacts on body patterning. All of the defects that I had talked about before determines where different tissues grow, how they organize, and it's a result of this one mutation altering the localization of this one protein in a key cascade for development and regulation. If you're interested in some of these things, Professor Saucerman in our own department has had projects involved with protein localization and traffic. And then in cell biology here, David Castle is interested in some of that membrane recycling, protein sorting that I talked about earlier. Questions on protein trafficking or localization as I switch over to the next topic? All right. if I start writing again, I'll put the lights back on. For the next 2.5, 3.5 lectures, we're now going to make a rather profound shift from central dogma, gene regulation, getting from DNA to proteins, to addressing where all of the energy for these processes come from. All right, up to this point, we've taken as a given that there are abundant energy sources inside the cell. This is true, but we haven't touched at all on where those energy sources come from or how they're generated. And that's what we'll be focusing on for the next week and a half. Our wrapper is obesity, excess body weight, and I think that this is known, but it has strong associations with many prevalent diseases in the Western population. Adult onset diabetes, type 2, you know that from the Modi lecture. High blood pressure, hypertension, stroke, blood clots in the brain. Hyperlipidemia, this is high circulating fats and triglycerides in the bloodstream, leading to things like atherosclerosis, heart attacks, as well as some cancers. The definition of obesity is defined with respect to what's called an ideal weight, so 20% larger than that. And the ideal weight clinically is defined by this metric called the body mass index, which is effectively a ratio of one's weight to surface area, so you see height squared, somewhere around there, trying to get a sense of how big the individual is. And so there are recommendations and scales on what these are to get a sense of what the normal or clinically normal range is, as opposed to obese in the numbers here. The seminal study showing the health detriments of prolonged obesity was the clinical study that I'm showing here from Scotland 20 years ago, plus years ago. Individuals were on a diet exercise regimen to lose 10% of their body weight and had profound impacts on overall lifespan. I already talked about hypertension, impacts on blood pressure. I already talked about adult onset diabetes, so better control of resting blood glucose levels, as well as reductions in hyperlipidemia, I said from before. Because of all these things, and when we get later on in this lecture series, when we talk about diets and things, and you get a sense of how difficult it is for one to shift their body mass index, change around their metabolism, it's come to be changed to obesity being recognized as a condition or a state, and more appreciated as a metabolic disorder, dysfunction, or something that either needs to be done both by lifestyle changes as well as potentially pharmacologic interventions. We have many objectives in this set of lectures. I'm presenting these in the order that we're going to try to accomplish them. At the beginning, I'm going to give you the fundamentals of metabolism centered on the generation of ATP as the key energy currency inside cells. I'll try to recall how, when the time is appropriate, to mention how things like GTP and other nucleotide triphosphates are generated in the cell, but the lecture will be very focused on ATP, which is for a good reason because the cells use it all the time as an energy source. In the setting of metabolism, we'll also talk about how fats are both synthesized and catabolized in the cell as an energy source. The combination of those two things will enable us to talk about different diets and how they are intended to work. And this will allow us to segue into metabolism of alcohol for the college students that are in my class. And finally, we'll close up with touching on the physiology of free radicals, reactive oxygen species, because these tie intimately to to metabolism for reasons that will become clear. And in fact, we had a big lecturer on reactive oxygen species earlier today. ATP. ATP is the fundamental energy currency in both prokaryotes and eukaryotes. You've seen the chemical structure before: triphosphate, ribose sugar, adenine base. Where the energy comes from for an overwhelming majority of chemical reactions is through hydrolysis of the gamma phosphate on ATP to generate ADP and inorganic phosphate. This hydrolysis reaction is highly exergonic. I think I have that on another slide coming up. Delta G, very negative, very thermodynamically favorable, and can be coupled with other kinetic thermodynamic processes in the cell to get you over the thermodynamic hurdle, enable things to occur by coupling them with ATP that wouldn't occur or wouldn't be thermodynamically possible otherwise. And we talked about many different biological processes that involve ATP hydrolysis: kinases, pumps. Right? What we haven't talked about is the backwards reaction. How do cells take nutrient sources and establish millimolar concentrations of ATP in cells all the time? 1 to 2 millimolar intracellular ATP are the common estimates of the concentration that cells have in them. If ATP is the key objective for metabolism to sustain life, to enable all of the processes that we've talked about up to this point in the semester, it would be advantageous to be able to synthesize ATP under different environmental conditions. And the key distinction of environmental conditions over evolutionary time is whether or not you have oxygen. If a cell, a tissue, doesn't have any oxygen, or oxygen is deficient, this is anaerobic metabolism. Anaerobic metabolism starts with substrates, nutrients, that are what are called fermentable, and converts them, depending upon the organism, to either lactic acid, lactate, or ethanol, along with carbon dioxide, to, in the process, convert ADP plus inorganic phosphate to ATP. And then that ATP circles back around through work, motors, okay, pumps, kinases, all these things. If oxygen is around, eukaryotes have a much more efficient way of generating ATP through aerobic metabolism, using the oxygen that's in the environment together with substrates that are oxidizable to generate water and carbon dioxide and boatloads more ATP, which once it's been generated can be used just the same as ATP generated by anaerobic processes to do work inside the cell. And even though we're not anaerobic organisms, we need to breathe, we do have tissues that will experience anaerobic conditions. The notable one is your heart. During vigorous exercise, it will go anaerobic. You could have skeletal muscle be exerted to the point of local oxygen depletion. So all of these anaerobic processes do take place in our body as well. They're just not chronic. To be able to talk about either anaerobic or aerobic reactions, we have to revisit redox chemistry from what I hope you got exposure to in first semester inorganic chemistry. I heard one scoff over there. So we're going to assume no prior knowledge, but I'm going to move quickly. To remind, oxidation reactions involve loss of electrons. These are electron transfers, okay? And reduction reactions are gains in electrons. As I said, especially for aerobic respiration, metabolic substrates are oxidizable. They can lose electrons. When they get broken down, those lost electrons carry energy with them that can eventually be used to generate ATP. So an oxidation reaction, the other thing to say is that these oxidation reactions are thermodynamically favorable to varying degrees, and I will emphasize that a little bit later on what makes them thermodynamically favorable, negative delta G, exergonic, in a moment. But here's an example of one. I talked about alcohol for a moment. We'll show it here again. Ethanol in our bodies is metabolized through an oxidation reaction that converts that ethanol to a byproduct called acetaldehyde. And as a result of that acetaldehyde, 2 electrons are lost from ethanol. That's the oxidation. Oxidation is lost. And those will get transferred elsewhere inside the cell. More to come on that in a moment. You'll note in this chemical reaction that even though it lost 2 electrons, the charge didn't change on the species. There are protons also that got released as a result of this chemical reaction, of this oxidation reaction, also get transferred to other species that play a role in the chemical reaction. But loss of electrons also means loss of hydrogen atoms, which is why in biochemistry, these oxidation reactions are frequently called dehydrogenation reactions, because they're taking off the hydrogen. It's a function of whether you're keeping track of the electrons and the hydrogen atoms come along for the ride, or you're keeping track of the hydrogens and the electrons come along for the ride. If this is a dehydrogenation reaction, by extension, the enzyme that catalyzes this reaction would be called a dehydrogenase. So alcohol is metabolized into acetaldehyde by alcohol dehydrogenase. Got ahead of myself. What happens to those released electrons during a dehydrogenation reaction, during an oxidation reaction? In aerobic metabolism, way down at the end, 2 classes from now, right, the final place for those electrons to go are onto oxygen with 2 hydrogen atoms to make water, H2O. But this is only at the very, very end of all of the things that I haven't told you about yet. Along the way, there are a category of biomolecules called coenzymes that, in this setting, what the coenzymes do is they serve as electron acceptors. They get reduced during that oxidation reaction, and then they can redonate those electrons later on in metabolism. I may call those reduced coenzymes reducing equivalents, and what it's meant to convey is that these, if you will, are stocking up the energy from the dehydrogenation reactions that they participated in, and then the cell elsewhere in metabolism will cash those in to make ATP, but it's later than in the first processes, the first dehydrogenation reactions that I'll present to you. Coenzymes generally are biomolecules that work along with the enzymatic reaction and can undergo changes in states, but they're not used up as a result of the chemical reaction, the dehydrogenation reaction. So it can change to a state, it can go from an oxidized to a reduced state, but it doesn't change into something else. I should also distinguish coenzymes from cofactors. Recall these all the way back from enzymology, magnesium or copper for certain ECM modifying enzymes. Those are usually just ions that you need to have in the active site of an enzyme for catalysis to occur. These are important for the chemical reaction. You need them, but they're not the same like a cofactor, which is just they're bound to the enzyme constitutively. And the first coenzyme that I'd like you to be aware of is called NAD, nicotinamide adenine dinucleotide. Just know NAD. Right? NAD exists in oxidized or reduced states. When it is in its reduced state, it's NADH. And NADH is what I'll refer to as a reducing equivalent. What that means is that NAD has picked up 2 electrons, picks up a proton to arrive at this reduced state. Here's an alternative depiction of the same thing if you're keeping track of the hydrogen atoms rather than the electrons and protons. It's in this way that NADH is storing electrons released from that oxidation reaction. Chemically, what does NAD look like? Has a lot of things that you've seen before. Adenosine. There's a pyrophosphate bridge here. Sugar, ribose sugar. The only part that you would have not seen before is this nicotinamide group. It's the nicotinamide where the redox reaction occurs. The oxidized form is as follows. When it gets reduced, these conjugated double bonds reconfigure. It picks up the hydrogen as shown here. And this is the reduced form with 2 stored electrons. Parenthetically, for those nutrition people, vitamin B3. Vitamin B3 is also known as niacinamide. Yeah, niacinamide. Niacinamide is a precursor to conjugate these nicotinamide groups. So that's why it's a vitamin. You'll see it in your cell culture. You should get it through your diet. You need it to be able to synthesize this coenzyme. We will see NAD in action very shortly. Let's talk about the first set of pathways that do not require oxygen. These are anaerobic pathways. 2 main steps. Before I even talk about the steps, I should say what the input is. What's going to start this whole thing off? Glucose. We saw other sugars in the setting of the lac operon, breaking down disaccharides to make monosaccharides. We talked about the importance of controlling blood sugar in the setting of insulin. Glucose is king. All right, we need this, and the reason why it's king is because it starts at the very top portion of this metabolic pathway called glycolysis. Glycolysis, as the name implies, takes glucose and will break it down into smaller byproducts and yield some energy or energetic equivalent in the process. We will talk about how you can run this pathway backwards and put in energy to many of the same chemical reactions in the glycolytic pathway to synthesize glucose rather than break it down. That metabolic pathway is called gluconeogenesis. So gluco, new, genesis, creation. And we'll speak about why that would be beneficial in different physiologic circumstances. I already said that glycolysis is anaerobic, so no need for oxygen for this pathway to take place. And where the aerobic versus anaerobic split occurs is at the output and the intermediate steps that happen after the output of glycolysis. And we'll see if we get to that point today. If those outputs of glycolysis begin to accumulate and there's no oxygen around, then those byproducts will begin to ferment. And that's the continued catabolism, the metabolic breakdown of those outputs, to generate a little bit of additional energy, but mostly to handle the byproducts so they don't build up and become toxic inside the cells. Glycolysis. I'm going to spare you the torture of having to memorize every enzyme and every intermediate species. This is the book, if you took regular biochemistry, that you'd be asked to commit to memory for 15 minutes and then it's forgotten as quickly as you memorize. The only person who knows this is me, because I have to teach it every year. What I'm going to try to do is emphasize the high-level organizing themes of glycolysis in the hope that those things stick. You will get quizzed on those, so please pay attention. Let's give a little bit more molecular detail on the inputs and outputs of glycolysis. As I mentioned, we start with glucose. Glucose is a 6-carbon sugar. It can exist in a ringed structure, like shown here. 1, 2, 3, I'm counting backwards. 1, 2, 3, 4, 5, 6. This ring can also open. There's an isomerization. You can just draw a cartoon and you get an open ringed form. That's shown on my textbook scan in here. 1, 2, 3, 4, 5, 6. This open chain version is a bit more tractable to appreciate the different carbons and how they're moving and when things get broken apart and so forth. But what I want to get across is that the ringed versus unringed is chemically identical. It's just for convenience of display. The output of glycolysis is to take that one 6-carbon chain and generate 2 3-carbon molecules of a metabolite called pyruvate. Pyruvate is centrally important to metabolism in the cells as the output of glycolysis and an input to many other metabolic pathways inside the cell. And so we're going to get to pyruvate and then we'll talk about the things that can happen afterwards. Glycolysis itself, we're going to organize into 3 basic steps. First is preparation of the glucose for it to be able to yield some energy to the cell, and also to cut it. As I said, we have to get 2 3-carbon molecules at the end. That's going to entail taking the 6-carbon molecule and splitting it in 2. After that, we have an oxidation reaction, a dehydrogenation reaction involving NAD. And then the third phase is the formation of pyruvate, which creates the output of glycolysis. Preparation and cleavage. The preparation of glucose and these early steps might strike you as a bit puzzling because they involve hydrolyzing ATP rather than synthesizing it. The high-level mass balance is shown up here. We're going to take glucose and hydrolyze 2 ATPs to generate this intermediate molecule, which I'll speak more about in a moment, and then we have the 2 ADPs as mass balance. What occurs? Here's glucose, open chain conformation. Step number 1 is to phosphorylate it through an enzyme called hexokinase. It's a hexose sugar. It phosphorylates it on one end here to generate glucose 6-phosphate. That's the first ATP hydrolysis that takes place. You have an isomerization reaction, which essentially means just shuttling around bond positions in a thermodynamically possible way. Moving the double bond here into the interior position to get you another intermediate, fructose 6-phosphate. There's another kinase that phosphorylates fructose 6-phosphate. Again, it's called phosphofructokinase, such that now there are phosphates on either side here. Fructose 1,6-bisphosphate. You can start to see some symmetry in this doubly phosphorylated molecule. There's phosphates on either end, 6 carbons in the middle. There's some double bonds and other things there. They don't quite line up, but they're starting to look close to one another. And then there's a cleavage reaction catalyzed by this enzyme called aldolase that splits the molecule in 2, these 2 byproducts here. Don't worry about it. Those two are close enough that one of the products can be isomerized to the other. So two phosphorylations, cleavage, and an isomerization to render two molecules of this metabolic intermediate called glyceraldehyde 3-phosphate. Write it in shorthand, G3P, but glyceraldehyde 3-phosphate. This is the first byproduct, two-molecule byproduct of glycolysis, because you split it in two. So that preparation is really to get glucose and its downstream metabolites ready so that when they're cut into, that you can generate two molecules that are identical to one another, biomolecules that are identical to one another. Loss of energy. So we're down 2 ATP so far. Next phase will be the first point of ATP generation, as well as the first oxidation reaction. From this point forward, we're just going to keep track of one of those glyceraldehyde 3-phosphate molecules, and I'll remind again and again and again that all of this stuff is happening 2 times. But we're only going to show one. Glyceraldehyde 3-phosphate is now oxidized by an enzyme that should not surprise you, is called glyceraldehyde 3-phosphate dehydrogenase, or GAPDH. GAPDH dehydrogenates glyceraldehyde 3-phosphate. And where do those electrons go? The electrons go onto NAD, generating one reducing equivalent of NADH per glyceraldehyde 3-phosphate. And I'll remind you that there are 2 molecules of glyceraldehyde 3-phosphate. So you've generated 2 NADHs. Bad job turning here. Sorry, I'm distractible. I can't talk over noise. All right. After that dehydrogenation reaction, we have the first generation of ATP. This occurs by directly transferring an inorganic phosphate to ADP to generate an ATP. And in fact, this type of phosphorylation, which is called substrate level phosphorylation, is catalyzed by a kinase called phosphofructokinase. And what it's an example of is a kinase getting run in reverse. If the concentrations of the products and reactants are such, and the activation energy barrier and the thermodynamics all works out, by shifting the concentrations of the two, you can have the kinase synthesize ATP by substrate level phosphorylation rather than hydrolyze it. And that's what occurs here 2 times. Those 2 ATPs have neutralized the 2 ATPs that were hydrolyzed during the substrate preparation step. And it's a bit enigmatic at this point, but if you'll take my word for it up to what we've said thus far, those 2 NADHs, those 2 reducing equivalents, have energetic value. We just don't see them yet in ATP terms, but we will by the end. So we have made some energetic equivalents that can be useful for ATP generation in the cells, but we don't immediately see it right now. Let's talk about the final step. I have a reminder. We're going around the bend here, so we're going to start on the right-hand side. The output of the oxidation reaction is a 2 molecules of 3-phosphoglycerate. There is a mutase that shuttles that phosphate around to the interior position. And then there's an enzyme called enolase that will do chemistry on this bond here to generate an enol group and a metabolic byproduct called phosphoenol pyruvate. There's a release of a water, but these are not generating or consuming any energy thermodynamically along glycolysis. And then the final thing that happens from phosphoenol pyruvate to get us to pyruvate is a second substrate level phosphorylation reaction catalyzed by another kinase running backwards called pyruvate kinase. Right, so the products and reactants, the phosphoenol pyruvate creates a driving force to synthesize ATP from ADP, an inorganic phosphate, and yield the output of glycolysis, which is pyruvate. And all of this times 2. So 3-phosphoglycerates, 2 ADPs converted to 2 pyruvates, 2 waters, and 2 ATPs. Where are we at the end? Here's the whole mass balance of glycolysis. 1 molecule of glucose, 2 oxidized NADs, proton balance, inorganic phosphates, and ADPs, generating 2 pyruvate molecules, 2 NADHs, reducing equivalents, proton and water balances, along with 2 ATPs. Seems like a lot of work to generate 2 ATPs from 1 glucose. What I want to relay to you now is that we are not done by a long measure with fully metabolizing these carbon sources. There's a lot more energy to be gained by continuing to metabolize pyruvate, and there's a good amount of ATP to be generated from those 2 reducing equivalents. But it's at this point, after glycolysis has occurred, that there's some decision making for the cell to do about how to handle the pyruvate after it's been spit out of glycolysis. And here's where the presence or lack thereof of oxygen is critically important. Here are the different paths that pyruvate can take after glycolysis. First, we're going to touch on briefly what occurs when there's oxygen around. In the majority of our tissues, this is what occurs. Pyruvate here is oxidized further through an enzyme called pyruvate dehydrogenase. Starting to see a trend here. It's this chemical reaction that is going to release one of these carbons in the form of carbon dioxide that we exhale. And it involves another coenzyme mediated reaction called coenzyme A. Chemical structure on that will be shown next class. It also generates another reducing equivalent of NAD, and its byproduct of this oxidation reaction is called acetyl-CoA, acetyl coenzyme A, that will feed into a metabolic pathway called aerobic respiration. And it's called several other things, but I'm not going to tell you what they are right now. Great. Is it going to start to, like, as if I was not already bothered by the ambulance? All right, I'm just going to keep going. Because I want to talk about fermentation. All right. What happens if there's not oxygen around? You can't have just pyruvate building up inside the cells. It needs to go via a pathway. And the fate of pyruvate after glycolysis depends on what organism you're talking about. So first, let's talk about yeast and other microorganisms. They ferment to make alcohol, which is why you put the wine and in beer and all these other things, right? Under the right conditions, they will take pyruvate, meaning no air. They have an enzyme called pyruvate decarboxylase to release a carbon dioxide. These are the bubbles in your beer or in your champagne, and generate a metabolic intermediate called acetaldehyde. Another oxidation reaction through acetaldehyde dehydrogenase. This consumes a reducing equivalent of NAD, but generates ethanol. All right, so this is the path that occurs in yeast microorganisms. It's not the normal route in humans because we don't have pyruvate decarboxylase. We can't do this first step. I looked around. I think fish might be able to do it, but we can't. Instead, we handle pyruvate via a different process, a different dehydrogenase. And the alternative dehydrogenase is called lactate dehydrogenase. Like this dehydrogenation reaction, this consumes a reducing equivalent and then generates the metabolic byproduct lactate or lactic acid, which is the best end stage metabolite that we can produce to handle the pyruvate load under anaerobic conditions. Now the lactate, you might have heard of the lactic acid buildup. Oh, you work out too much, muscles are sore, or all these other kinds of things. Right? It can build up in the heart. It can build up in skeletal muscle. We physiologically need to have mechanisms to be able to handle this lactate and process it. And this invokes a coupling between two different tissues, organs in our body, and the coupling of glycolysis, the breakdown of glucose, like I just talked about, and gluconeogenesis, the synthesis of glucose from the precursors, the metabolic byproducts of glycolysis. And the cycle between this glycolysis that's occurring in the skeletal muscle and gluconeogenesis, which occurs in the liver, together is called the Pore cycle. And it's meant to, this is involved during recovery after exercise. And it's has blood as the communication channel between these two tissues. I already talked about the process on the left. Glucose, lactate, not enough oxygen. That's how you got the lactate. Lactate is diffusible. Right? There are transporters and things in cell that gets excreted from cells into the bloodstream. The liver takes up lactate and will now put energy in to converting the lactate back to glucose. So it's energetically costly, but it's better than acidifying the cellular environment. And you need to be able to deal with this waste byproduct. And this is what hepatocytes, the liver cells, do. They'll take it in. They're going to the extent possible, they'll generate some reducing equivalents, get you back to pyruvate, put them back in. They're going to use a good number of ATPs to be able to get you back to glucose. But now you have a metabolizable substrate that any cell in the body can use. This is the essence of the Pore cycle. Which is a nice transition for us to do the compare and contrast between glycolysis and gluconeogenesis. First message is that there are a lot of similarities between the two. Many of the steps that I touched on where it's like, oh, it's energetically neither here nor there, it goes back and forth, those are identical between glycolysis and gluconeogenesis because the thermodynamics of whether the reaction runs in the forward direction or in the reverse direction is entirely dependent upon the concentration of substrates and reactants. If a cell is set up that there's more products rather than reactants, then that reaction will run in the reverse. However, there are 3 steps in glycolysis that are thermodynamically irreversible. I mean, you can't jigger the concentration of products and reactants to be able to have the reaction run in reverse. And this is because the delta G of the reaction itself is so large that there's no way you can offset it physiologically. Those 3 irreversible steps occur at the top and at the very top and at the very bottom of glycolysis. The 2 initial phosphorylation steps, the 2 ATPs that were consumed during the substrate preparation, you can't go back and make ATP by those processes. To remind, those are catalyzed by the enzyme hexokinase and the enzyme phosphofructokinase. So phosphate 1, phosphate 2. Once those phosphates are on there, the only way that gluconeogenesis can get you back to glucose is to chop off the phosphate. So there are, can you read that? You probably can't read that very well. Those are phosphatases that simply hydrolyze the phosphate on glucose 6-phosphate or fructose 1,6-phosphate to get you back to the non-phosphorylated species or less phosphorylated species. Those are the 2 steps at the top. The step on the bottom is that substrate level phosphorylation involving pyruvate kinase. You can't run that in reverse either under cell physiological conditions. And what gluconeogenesis in the cell does is bypass pyruvate through this metabolic intermediate called oxaloacetate. Keep that in your mind. It will come back later. It's in another metabolic cycle that I haven't talked about yet. And by virtue of putting some ATP into the system and even some GTP hydrolysis events, this will get those carbons back to phosphoenolpyruvate, and then many of the other steps in the middle can be run in reverse by virtue of products and reactants. We saw gluconeogenesis involved in the Cori cycle, so handling of lactate, circulating lactate. There are other reasons and evolutionary advantages to running glycolysis in reverse. Another one is what happens if you just have a lot of glucose, or if there's a lot of circulating glucose, you generate a lot by gluconeogenesis, but you don't need to metabolize it right in the moment. How does the cell handle that excess glucose to not waste it or just have it sit around? Is there a way to store it in a particular way to mobilize it rapidly on demand, even if circulating blood glucose levels might be low? And so this can occur in just times of feast when there's a lot of circulating blood glucose or active gluconeogenic activity. This occurs a lot in the liver. And what the cell will do is store, if we talk about ATP as the currency, this is like put money in the bank. Right? It'll store glucose for future needs as a long-chain branched polymer called glycogen. And what glycogen acts as is as a condensed repository of polymerized glucose that can be mobilized very rapidly for glycolysis. Muscle will also do this. How does this occur? Glucose 6-phosphate is the second step down from glycolysis. It's after that first phosphorylation event by hexokinase. So a phosphate on the 6th position very quickly can be moved over to the 1 position on the carbon by one of these mutases. So just shuttling around. It's not an isomerization reaction, but you just shuffle the position around to make glucose 1-phosphate. Glucose 1-phosphate is modified with by a UDP. I was going to call that UDPylation. I've never heard that before. So it picks up a UDP to become a UDP glucose using UTP, so uridine triphosphate. And this UDP glucose acts as a monomer and a substrate for a polymerizing enzyme called glycogen synthase. Glycogen synthase will take either glucose, UDP glucose monomers or a UDP glucose monomer and a growing glycogen polymer and tack on a glucose onto one of these chains. These are 4 linkages here, long linear chain. As the formation of glycogen continues to proceed, there are other oxygens here where you can branch off additional sugars, additional glucose molecules through so-called 1,6 linkages. At the end, a fully formed glycogen particle inside the cell, let me check my numbers here, can contain 50,000 glucose molecules in one macromolecular polymer. So it's a very dense way of storing glucose inside the cell for future need. And it involves taking a detour off of glycolysis after the first step and running it into this polymer store. When there is a need for glucose rapidly, in muscle, for example, the glycogen gets broken down. There is an enzyme called glycogen phosphorylase that will transfer a phosphate to the glycogen polymer, and it's called a phosphorylase because it's the direct transfer of an inorganic phosphate rather than through ATP. Otherwise, it'd be called a kinase. moves that inorganic phosphate, attacks the glycogen polymer, and will release a glucose 1 phosphate, which is readily reconfigured by that mutase to get you to glucose 6 phosphate, which then puts you right back into glycolysis, where I took you away for glycogen synthesis. Therefore, by rerouting these metabolites into a storage form and then mobilizing them rapidly, tissues that are very metabolically active have this store of material. I have one more slide, and I know I have 2 minutes. Hear me out, because otherwise it's post-lecture. Last thing, everything that I told you up to this point, we talked about glycolysis, gluconeogenesis, irreversible steps in glycolysis, and how gluconeogenesis has to work its way around those steps to be able to get to the final byproduct. What I haven't talked about are the way in which glycolysis is controlled internally based upon the metabolic state of the cell. There are multiple points of control in glycolysis that dictate how rapidly or effectively each one of those metabolic reactions occurs, depending upon other byproducts in the cell. And the 3 places that it occurs are at the 3 irreversible steps that I talked about from before, which makes sense, right? Once you've gone over the point of no return there, it's costly to go backwards, right? And so those ones should have an additional layer of control. Here are the 3 examples. Hexokinase, its activity is negatively regulated by the concentration of glucose 6-phosphate. So the product of the enzyme reaction that it catalyzes, if it's too high, it feeds back and it inhibits the activity of the enzyme. Negative feedback. There's another negative feedback at the level of phosphofructokinase. It's inhibited by the concentration of ATP in the cells. So if metabolism and glycolysis is humming away, there's no need to continue metabolizing glucose. It should be getting routed to glycogen stores. And therefore, if ATP concentrations are high, feeds back negatively on PFK activity, shut down the pathway. And then the final example is not a feedback regulation. It's a so-called feed-forward regulation, because it's a metabolic precursor informing that downstream enzyme, the last step of substrate-level phosphorylation, pyruvate kinase. Its activity is promoted by this metabolic product here, fructose 1,6-bisphosphate. In many ways, you can think about this as glycolysis, the tail end, getting ready for the byproducts that are going to come down the pike, all right, to maximally convert phosphoenolpyruvate into pyruvate and then route it to the other metabolic points. And so what I wanted to have stick with you is that these are convenient and effective control points because they're coupled to the irreversible steps in glycolysis. I will stop. Thank you for sticking with me. Have a nice weekend.